Hi everyone, my name is Farrah Leone. I'm the lead technician on the South Brook Wind Telemetry project. We just finished the pre-construction phase of our work, and that's what we're going to be telling you about today. So let's get everyone on the same page here. The blue circle you see is all the area being leased for wind farm development. Our wind farm is in the bottom left corner. The blue line that runs from the windmills to the X is the export cable, so it's taking all the power to the grid. The X you see is where our study site is, and also where it will be connecting to shore. That is Wainscott, New York. East Hampton, for anyone that's not familiar with the area. So the wind farm is 35 miles east of Montauk Point. It's going to be 15 turbines, and it's going to power 70,000 homes. So what our lab is studying is the presence and impact of electromagnetic fields on electrosensitive species. So some of the species that we are looking at include striped bass, summer flounder, black sea bass, and different local Latin spring species, but there's also some species, some auxiliary species we're looking at, such as sturgeon, American eels, and Atlantic cod. So our project goals are, like I said before, to evaluate EMFs, electromagnetic fields, on behavior and movement of targeted species pre, during, and post-construction. So we just finished the pre. And to estimate movement metrics such as acceleration, depth, 2D and 3D position, and residency for telomered individuals. So how are we going to do that? We currently have 43 acoustic receivers out specifically in that Wainscott site. So if you look at that little turquoise tip at the top, that is the acoustic release receiver, and it is connected to the flat black line with the two floats. That's bolted together. Underneath that is a canister that's filled with coiled rope. All of that now stays together by a lug at the bottom of the canister. It's very light, a little hard to see in this photo, but where you see the gray tip before it connects to the red line, there's a lug. So me or someone else on the team will be on the boat with a VR100. We put a hydrophone in the water and we send a signal that allows the lug to unscrew itself and it's caught to the surface. And we get everything back because everything is attached with that rope. And in order for our receivers to get anything, we need to tag fish. So, as Maria and Ashley were saying before, the really big one you see is called a V16. We put that in our larger species and all the sharks. In the middle, we have a V13. V13s we're going to put in our skates. What's also special about some of the V13s that we have is we have them able to detect acceleration and pressure for specifically striped bass. So we can figure out where the water column they are when we get the data back. And the smaller one you see in the end is called a V9. We're going to put that in smaller species such as summer flounder. And the way that basically this works and how I've heard it described to me is that it's like an easy pass. The fish goes by the receiver and the receiver collects the time it went, the number associated with the tag inside of it, and the date. So this is the actual array. All of the yellow dots are our receivers. There's 43 in the entire array, and we have a fine scale array, which you see everything really clumped together up there in the middle of the cable approach. And the blue dashed line you see is the projected area of where the export cable is going to be, so that's where theoretically the electromagnetic field is going to be the strongest. It runs directly through the fine scale array, and underneath one of the real-time buoys represented by the white circles you see above. And the real-time buoys are really cool in theory because they actually will send technicians live data when a fish swims underneath them. So right now, here are all the active tags between all the labs that Soma's working on this project and Monmouth University across a ton of different species. As Ashley was talking about before, been tagging sturgeon for 12 years, so we have a lot of those right now. And striped bass is a big commercially important species for this project. We're tagging about 40 of them a year, so there's a few hundred that will be tagged by the end of the entire project, which is projected to be about 2025. And now for detections by species, clearly we have a lot. And little skate really overtakes every other species, but there's even hundreds of thousands of detections for some of these other species, which the little skate really seems like it destroys. But if you look at black sea bass, you look at the clear-nosed skate and tautog, there's still 100,000 detections. Silver flounder's not that far behind. We even have a few thousand detections for sharks. So we have been able to identify 44% of all the tags that have came in across 19 different species. And although we're having some success with every project, there's going to be some issues. First issue, if you couldn't tell by my blog before, was the real-time buoy. That one right there. We're using it on another project, and there's probably been like a year and a half of troubleshooting because we are one of the first groups to be using this equipment and using it in saltwater, using it in the ocean. It's taking a little bit of an adjustment period to get it to work. So we are planning to put them out in the spring, which is really exciting. So my phone's going to be blowing up all the time with different fish. And then if you look at the map, so I've gone out 3 total times to switch receivers. And this map shows you the amount of times that I have gone out and a receiver has been missing from where I last put it. And that doesn't mean that it's gone forever, but that just means that it's not where I last put it. Sometimes we get them back months in advance, sometimes we get them back before the swap even happens, but sometimes we have problems keeping them where they're supposed to be. But even though sometimes we're missing our receivers and the real-time buoy is not always working, we're getting a lot of data. So this is the fine scale array with the 20 close-knit receivers from the other map. Every single individual color on this graph is a different individual. Like individual, not an individual species. These are all different animals, and there's so much movement being detected, you can barely see the receivers through this. What's even more special about this picture is that the export cable runs directly in the middle of it. So if something were to happen, if the magnetic fields are really impacting the animals, we're going to know immediately. There's going to be a direct line down the middle of it if something were to go wrong. So I want to compare these two movement patterns specifically, because that's the main reason of the fine scale array, is to show how animals are utilizing the space and to compare it to during and post-construction. So I talked about earlier about how the striped bass had those tags that accounted for acceleration and pressure. The larger the bubble, the higher it is to the surface, so the more shallow it is. And with striped bass, we're seeing that they're kind of just going straight through the area, at least from the data we have now. They're not hanging out, they're not spending a lot of time there, but then you go and you look at the little skate, and they seem to love it. They're going in circles, they're not leaving, some of them are leaving and coming back. So it's really interesting to see how the different species utilize the same area. And although there's not too many individuals that look like they're being picked up here, there are some species that are giving us an abundance of data, and this abundance of data can tell us really important things about the habitat. So for example, if you look at I know there's a lot of pale yellow, but the palest yellow lines, you see kind of swerve in and out of different parts of the array, which could tell us that this space is at least important for this individual, whether it just likes the area or if it's hunting or whatnot. We're going to find out as time goes on. But for now, we've only finished the pre-construction phase, and we have a few future objectives, which mostly encompasses just finishing the project. Big future objective for me personally is getting the real-time buoys out and making sure they work and they stay in place. Getting data for during and post-construction, and then our students are going to be comparing this data through models and to just continue tagging. And I'm probably going to be back here next year talking about what's happening during construction. I'm really excited to see what we'll be. Thank you.